Bismillah ar-Rahman ar-Rahim. This is the second lecture in Introduction to Medical Virology. It will be about viral replication. Viral replication, for the virus to replicate inside a living cell, there is a certain requirement for that. So the requirements for viral replication includes: 1. Living cell. Viruses multiply only inside the living cells, redirect host cell biosynthetic processes for synthesis of viral components. 2. Host cell must provide energy, synthetic machinery, low molecular weight precursor for synthesis of viral proteins and of course nucleic acids. Third requirements: viral nucleic acids. The viral nucleic acids, which carries the genetic code specific for all viral specific macromolecules. Therefore, the virus must be able to produce usable messenger RNA for successful viral replication. As a start, we have stages of viral replications. Stages of viral replication: 1. Attachment, or we can call it adsorption. 2. Penetration, also we call it viropexis. 3rd, uncoating. 4th, expression of viral genome and synthesis of viral components. 5th, assembly or morphogenesis and maturation. Assembly, morphogenesis and maturation. 6th, we have the release. On the right, this picture showing the different stages of viral replication. Started with absorption, then penetration, after that uncoating, expression of the viral genome and synthesis of viral components, morphogenesis, assembly, maturation and release. Now we will take each stage one by one. First stage: attachment, adsorption. Here we will have interaction of the virion with a specific receptor on surface of the host cell. Host cell receptor differ for different viruses, but generally they are glycoproteins. So host cell receptor generally are glycoproteins located on host cell. Some virus bind to protein sequence, protein moiety of this receptor. Example, pacornavirus. And on the other hand, other viruses bind to oligosaccharide moiety of the host receptor, such as in case of orthomyxovirus and paramyxovirus. The receptor binding reflect a configurational homologies between virus surface structure and the cell surface components. very important point. The presence and distribution of the receptor are important determinant in cell tropism. Or, in the other word, the cell tropism of the virus, it depends on the presence and the distribution of the host cell receptor. For example, a human immune deficiency virus binds to CD4 receptor on the immune cell, whereas Epstein-Barr virus binds to CD21 receptor on B cell. This picture showing the HIV virus with the surface glycoprotein 41 and the glycoprotein 120. The receptor for the HIV virus is CD4 and the co-receptor for the virus is the CCR5. Again, there will be binding between the viral glycoprotein 120 with the host cell receptor CD4. This represents configurational changes in the host receptor, followed by the process of using the co-receptor CCR5 in the starting of the replication of the HIV virus, followed by membrane fusion between the virus and the host cell membrane. Second stage in viral replication is the penetration to the host cell, or we can call it viropexis. The process by which the virus particle taken up inside the infected cell called penetration, engulfment. The virus can enter the cell by several mechanisms. The virus can enter the cell by several mechanisms. One, direct penetration. Only viral genome of the virus transfer through the plasma membrane. This is called direct penetration. The second process of the penetration of the virus inside the host cell is a receptor mediated endocytosis. In receptor-mediated endocytosis, there is a transfer of entire viral particle across the cell membrane by endocytosis, forming endosome. Third way of entrance of the virus inside the infected cell is the fusion of the virion envelope with the host cell plasma membrane. There is a fusion between the plasma membrane and the envelope of the virus, releasing the nucleocapsid inside the cell cytoplasm. So, in case of naked viruses, it can enter inside the infected cell, either direct penetration, whereas there is only the transferring of the genome of the virus inside the infected cell, or by a process of endocytosis, forming endosome. Whereas the envelope viruses, it can enter inside the infected cell by a process of endocytosis or a process of membrane fusion, where there is a fusion between the envelope of the virus and the cytoplasmic membrane of the infected cell. Third process of the viral replication is the uncoating. In the stage of uncoating, there will be removal of the capsid of the virus and core protein, liberating viral nucleic acid. This process is termed uncoating. With the exception of pox viruses, the process of uncoating utilizes pre-existing cellular proteases. Pox virus uncoating requires the synthesis of a new pox virus-coded proteases. By this process, virion disrupted and lost its infectivity. This phase of viral growth cycle is called the eclipse period. Its duration varies depending on both the particular virus and the host cell. This period is followed by rapid accumulation of infectious progeny virus particles. The fourth stage of viral replication is the expression of viral genome and synthesis of viral components. Here, the specific messenger RNA, a transcript from viral nucleic acid, for successful expression and duplication of genetic information. Then, viruses use cell components to translate the messenger RNA. Mechanism of transcription: either 1. Direct transcription of messenger RNA without intermediates like most DNA viruses, adenoviruses for example, herpes viruses as another example. 2. Viral nucleic acid serve as a messenger RNA. Example: this is an example in positive sense single strand RNA viruses like Pacorna viruses. Here, the nucleic acid of the virus is a positive sense, that is to say it's 5 prime 3 prime. Reading frame of this genome of the nucleic acid of these viruses is like the reading frame of the messenger RNA of the cell, so it's called positive sense nucleic acid. In case of positive sense single-strand RNA viruses like the coronaviruses, the nucleic acid of the virus serves as a messenger RNA. Third way of transcription, here the virion carry its own RNA polymerase to synthesize its own messenger RNA. RNA viruses of this type are called negative sense single-strand RNA viruses. Example, rhabdoviruses is an example of this group called negative strand viruses or negative sense single-strand RNA virus. Fourth way of transcription, here the virion contains reverse transcriptase in which viral RNA code for complementary DNA. Virion contains reverse transcriptase in which the viral RNA code for complementary DNA, like the case in retroviruses. Example, HIV virus. The intracellular site of viral replication, the viral protein is synthesized in the cytoplasm on the ribosome, composed of viral specific messenger RNA and host cell ribosome. DNA viruses, with one exception, replicate in the nucleus. Most RNA viruses undergo their entire replicative cycle in the cytoplasm. This picture represents the DNA virus replication. The virion first adsorbed to the host cell, then penetrate inside the cytoplasm of the infected host cell, go directly to the nucleus, as most DNA viruses replicate in the nucleus of the host cell through transferring to the nucleus, uncoating process taking place, releasing the viral nucleic acid. Depending on the DNA, dependent RNA polymerase of the host cell, the DNA of the virus, DNA dependent RNA polymerase transcript into messenger RNA, viral messenger RNA, goes to the ribosome of the infected cell to synthesize viral protein, and on the same time the nucleic acid of the virus, depending on the DNA polymerase, synthesize a new copies of the viral genome. After that, this will follow the process of morphogenesis, assembly, and releasing from the infected cell by a process of budding. In case of RNA virus replication, first we have the positive sense single-strand RNA virus. Here the genome of the virus translated in the ribosome to synthesize viral proteins, and on the same time there will be duplication of the genome of the virus to synthesize new copies of the viral nucleic acid. In case of single-strand negative sense RNA viruses, here the negative strand RNA transcript to form positive strand template for the synthesis of messenger RNA of the virus to go to the ribosome to synthesize viral protein, and this template serves as copies to synthesize a new nucleic acid of the virus. The fifth stage of viral replication is the assembly, maturation, and release. A newly synthesized viral genome and capsid assemble together to form progeny viruses. After formation of complete virions, the viral particles release from the infected cell. Mechanism of viral release from the host cell include host cell lysis or budding through the cytoplasmic membrane. First mechanism اللي هي host cell lysis referred to as the lytic cycle, which result in the death of the host cell. The non-enveloped viruses release in this way. In case of budding from the cytoplasmic membrane, enveloped viruses mature and release by budding process. The virus-specific envelope glycoproteins are inserted into the cellular membranes, and viral nucleocapsid bud through the membrane at these sites and acquire the envelope. Sometimes viral maturation is inefficient to process. Sometimes process of viral maturation is inefficient, resulting in excess amount of viral components accumulate, lead to the formation of inclusion bodies in the infected cell. Cells. This EM picture showing the process of releasing of a new progeny virus from the infected cell by the process of budding. This is a schematic diagram of whole steps in viral replication. First step, viral attachment adsorption, penetration, uncoating, expression of the viral genome, that is transcript to form messenger RNA, and at the same time replicate to synthesize new copies of the viral genome, followed by packaging by the maturation, assembly, and lastly the release. Here the release is taken by a process of budding, taking the cytoplasmic membrane of the infected cell at the site where it's decorated with the viral coded glycoprotein to form the viral glycoprotein projection on the surface of the envelope of the new progeny virus. Now we will come to the pathogenesis of viral disease. Steps in viral pathogenesis include viral entry into the host cell, primarily viral replication, viral spread, cell and tissue tropism, cellular injury, viral shedding, and lastly the host immune response. All these steps we will talk about each step quickly. Entry into the host cell or into the host. Some viruses enter into the host through the skin. Dead cells cannot support the virus replication. Most viruses which infect via the skin require a break in the physical integrity of this effective barrier, like there is a cut in the skin or abrasion. Many viruses employ vectors to bypass the skin, like arbovirus using the ticks, mosquitoes, or even vampire bats to break the skin barrier. Example of the viruses that enter through the skin is the human papilloma virus and rabies virus. Second way of entry to the host is through the respiratory tract. Again, the respiratory tract and all other mucosal surface possess immune defense mechanism like the secretory IgA, as well as non-specific inhibitory mechanism represented by the ciliated epithelium, mucus secretions, and the low temperature of the upper respiratory tract. So the virus must bypass all these barriers in order to enter through the respiratory tract. Example of the viruses enter through the respiratory tract is the influenza virus, rhinovirus, coronavirus, and etc. Other viruses can enter through the gastrointestinal tract. Gastrointestinal tract is a hostile environment for the viruses because of the gastric acid, bile salts. So again, the viruses should have ability to overcome this condition in order to enter through the gastrointestinal tract. From the example, or as example of the viruses that enter through the gastrointestinal tract, we have the hepatitis A virus, poliovirus, rotavirus. Other viruses can enter through genital tract. Example, HIV virus, human papilloma virus, hepatitis B virus, herpes simplex virus 2. Other viruses enter through the conjunctiva like the herpes simplex virus 1. Some viruses enter through the blood directly in case of hepatitis B, C, delta agent, HIV, or even the CMV through the contaminated blood or using a needle or other sharp object contaminated with the virus. Second process in the pathogenesis of the viral infection is the primary replication. Usually, the site of entry of the virus is the site of replication as well. Here, virus produce disease at the site of entry and have no systemic spread. Example of localized viral infection in respiratory tract is influenza virus. In the gastrointestinal tract is rotavirus. Localized viral infection in the epidermis, a good example of this is the papilloma viruses. Other viruses end by systemic infections. Here, at the site of entry of the virus, there is a primary replication, followed by spread to the blood to reach the main target, where secondary replication takes place. Enteroviruses, as example, enter through the intestinal epithelium, then through the lymphoid tissue associated with the GIT to reach the blood, and then to reach its main target, which is the CNS. Other example is the herpes viruses enter through the oropharynx or genitourinary tract, then through the local nerve ending to reach its main target, which is the CNS. Third point is the viral spread. Viruses can be directly transmitted or spread from cell-to-cell contact. Direct cell-to-cell contact. A part of this, we have two main mechanisms for viral spread: a bloodstream and nervous system. A bloodstream, the presence of the virus in the blood, is called viremia. Virus may get into the bloodstream by direct inoculation, in case of arbovirus by arthropod vectors, or by blood contaminated blood transfusion, intravenous drug abuse, or spreading from the site of primary replication. Virus may travel in the blood free in the plasma, like the case of toga viruses or enterovirus, or associated with the platelets in case of herpes simplex virus, or associated with the lymphocyte in case of the EBV and the CMV. Other viruses is associated with the monocyte in case of lentivirus. Other way of spreading of the virus is through the nervous system. In some cases, a neural spread is involved, like rabies virus reach the brain to cause the disease. Herpes simplex virus moves to ganglia to initiate latent infections. Cell and tissue tropism. Tropism is the ability of a virus to replicate in a specific cell or tissue. The ability of a virus to replicate in a specific cells or tissues. This organ and cell specificities reflect the presence of a specific surface receptor for the virus. Receptors, it is the components of the cell membrane with which the viral surface, whether the capsid or the envelope, can specifically interact and initiate infection. Example, HIV bind to CD4 of T cells. Poliovirus bind to gut epithelium, in neurons, in spinal cord and brain. Cellular injury. Viruses can cause cellular injury by destruction of the viral infected cells due to the viral replication or due to the immune response to virally infected cells in the tissue are responsible for the development of clinical illnesses. Destruction of virally infected cells, whether due to the viral replication or due to the immune response to the virally infected cell in the tissue, this is responsible for the development of clinical illnesses. As a result of viral replication, cellular cytopathic effects develop and cell death. Sometimes cell is not damaged by the virus and result in persistent infection. Some tissue, like intestinal epithelium, can rapidly regenerate and withstand excessive damage better than other, like the brain. Sometimes, as a result of viral infection, there is a transformation of the infected cell. Later on, can lead to neoplasia. This kind of viruses is called oncogenic virus. Sixth point in viral pathogenesis is viral shedding. It represents the time at which an infected individual is infectious to contacts. Viral discharge into the environment to maintain a viral infection in population of hosts. Shedding usually occurs from the body surface involved in the viral entry. For example, influenza virus enter through the respiratory tract, and the shedding of the virus occur also with the respiratory discharge through the respiratory tract. Or in case of rotavirus, enter through the gastrointestinal tract, and the shedding of the virus again occur through gastrointestinal tract with the infected stool. Herpes simplex virus 2 enter through genital tract, and the shedding of the virus again occur with contaminated genital secretion. Shedding of the virus occur at various stages of disease depending on particular agent involved. Now we will come to the host immune response to the viral infection. Host immune response to the viral infection has a major impact on the outcome of the viral infection. The most prominent is the induction of interferons, which induced soon after viral infection. Interferons inhibit viral replication before specific humoral and cell-mediated immunity begin. Virus-encoded proteins serve as good target for immune response. The virus-encoded proteins is a good target for immune response of the infected host. Virus-infected cells lysed by cytotoxic T lymphocyte. A humoral immunity protects the host against reinfection. Neutralizing antibody block the initiation of the viral infection. Again, a humoral immunity against the virus protect the host against reinfection. This is by that the neutralizing antibody block the initiation of process of viral infection. Neutralizing antibody coding or bind to the viral attachment to protein prevent the first step in the viral infection, which is the adsorption attachment, where the viral binding protein coated by the antibody, it will not be recognized by the host cell receptor. So by this process there will be aborting of the process of viral replication in the first stage. Thank you for your listening. This is the end of the second lecture in Introduction of Virology. شكرا جزيلا.